Everyone, welcome to our 8th of June 2026 webinar for the UK Freshwater Quality Research Programme. My name is Joseph Holden, and along with Pippa Chapman, I'm forming part of the Programme Champion team for the Freshwater Quality Programme. Currently, the programme has 6 projects within it, the most recent of which is at the top there, called ClearNay, and that's looking at Loch Ney. We have a project which is Yorkshire-based called EcoMix, Pacific, which is on chemicals and mixtures and the ecological impacts. And then we have MOT for Rivers, which has a focused catchment in Scotland, but a wider set of data sets and monitoring that's been going on and modelling going on. Quantum, which is looking at livestock impacts. And then the focus of today's webinar, which is LTLS-FE, and this is a fantastic modelling project looking at UK-wide implications for the future. So absolutely delighted to have the team here today to talk to us about the amazing progress on this project. So quite soon we've got another couple of project updates in our programme. So later in June we've got EcoMix, and then in July we'll hear from Pacific. So do add those to your diary, because of course the benefit of joining live is that you can interact with the presenters and ask questions, challenge on issues and so on. And we don't record that part of the process, so it allows for kind of free and frank discussion. It allows people to kind of open up a bit more. So we only record the actual presentation itself from each of the presenters, but then we kind of pause and stop that presentation when it comes to Q&A. So do please join live if you can for these events. There's a huge benefit in doing so. And of course, there are links to web pages and other resources and so on, including biosecurity e-training. And please do follow those links to find out more about the programme and more about how to be careful around biosecurity when you're out sampling freshwaters in the UK.
Great. And yeah, as I mentioned previously, at the start of this webinar, we have a very brief 2-question survey, the link of which you can use the QR code. I don't expect you to type out that URL, but it's in the Q&A section. It's a pinned link there. So yeah, this is there's an opportunity arisen for a seed corn funding opportunity with Ulster and Taiwan colleagues to develop a sensor that can potentially be used to detect contaminants, but we want to make sure that what is being developed is useful for the wider community at large. And what better way to ask and to understand what that ask is to maybe to ask you today. So yeah, if you could just list your top bacterial contaminant that you'd like a sensor to be able to detect, and one of your top PFAS type contaminants as well. That'd be really useful. And this will be live throughout the duration of this presentation. So yeah, please do fill that in, and we'll come back at the end just to sort of see what the kind of the word cloud looks like in terms of what people's priorities are. Thank you.
Thanks, Finn. So nothing else to say other than thank you all for joining, and thanks so much to our presenters. I'm just going to hand over now to the LTLS-FE team. Thank you. So, Vicky, you're muted. I don't know who's kicking off with presenting, but if you could unmute, please.
Morning, I think Steve is starting off. Steve?
Okay. So, welcome everyone. Thank you very much for coming today, and we're going to present an overview, a model summary, and some results from the LTLS Freshwater Ecosystems project so far. So I'm starting off with the overview, then our colleague Nathan will talk a little bit about the model summary and assessment. Vicky will show some material on future scenarios, and then we will talk about next steps and stakeholder engagement. So next slide, please. Okay, so this project developed out of a previous project called LTLS Long-Term Large Scale, which was to do with exploring how carbon, nitrogen and phosphorus fluxes in the UK river system has changed since 1800. And this basically set up a UK scale model operating at a resolution of 5 kilometers to make those macronutrient carbon, nitrogen, phosphorus flux predictions over the whole of the UK river system. And therefore, for this project, LTLS-FE, which is long-term large-scale freshwater ecology, we're using a similar modelling approach to quantify future projections of a broader set of freshwater quality variables, and also to model their impact on biodiversity within UK rivers. So next slide, please. So this sort of summarizes how our project is structured. In work package 1, we assessed and chose a set of dominant pollutants affecting UK freshwater health to work with and to incorporate into the modelling. You'll see some examples of those later on. We developed models of terrestrial soils to simulate the soil to freshwater transfer of our pollutants alongside the nutrients which are already built into the model. And we had a work package to gather together the driving data for our models, including historic and present day emissions of our pollutants into the freshwater environment, but very importantly, our future scenarios of chemical emissions and climate change, which are based around 6 combinations of climate change scenarios and also socio-economic scenarios, incorporating changes such as population, the amount of traffic on the road, social attitudes to the way that we use chemicals and things like that. So work package 1 is complete. What we're going to mostly show you today is our ongoing outputs of work package 2, which are the outputs of our integrated freshwater model, which takes all that information from work package 1 and integrates it into our freshwater model, and also our impacts on biodiversity. So that work is ongoing and it's feeding into our work package 3, also ongoing, which is analysis of outputs for stakeholder uptake and use, and our model and data dissemination, and our web dissemination, which we will talk a little bit about towards the end of this talk. So next slide, please. So some examples of the new stuff that we've done within this project. We've created a UK-wide sewage and urban runoff model that accounts for population, effluent production, and the capacity and efficiency of wastewater treatment, which may be projected to change under one or more of our future scenarios. Our colleagues at Rothamsted Research have developed a new agricultural pesticide model. We have produced data sets of UK-wide emissions of a range of chemicals that we consider to be the priorities to consider in terms of impacts, and those include metals, personal care products, industrial chemicals, PAHs, and pharmaceuticals, alongside the macronutrients. One little highlight of that, we have made UK-wide estimation of current metal loadings from abandoned mines. We have also produced a monthly abstraction data set for both surface water and groundwater, which contributes to the hydrological modelling that underpins all of our water quality modelling. And then finally, we have the UK Freshwater Quality Model, which has been tested and assessed using historical observations, and you'll see some more results of that later on. Next slide, please. A summary of what we've done. We've modelled over 75 determinants, ranging from water flow, pH, temperature, oxygen, fairly basic stuff, to macronutrients and micropollutants and metals. We've included over 40 new data sets in driving the model. We've got a mean seasonal performance model of, across 20 determinants of 0.44. We've made over 450 megabytes of model output so far. We've got between 1 and 200 observation sites available for each pollutant against which to assess the model performance. And as I mentioned earlier on, we've got 6 future scenarios evaluated from 2020 up to 2080. So next slide, please. Okay, and I think here I'm handing over to Nathan, who's going to take you through model summary and assessment against observations.
That's right. Yeah, I'll take it from here. Let's go to the next slide, please. So I'll quickly talk you all through our model a bit. So this slide shows a flow diagram of our modelling framework. The model is driven by several inputs, including land cover data. So the model gets fractional land cover data per 5 kilometer pixel, like shown in the top left there. In addition to climate data, soil properties, land management, and emissions from a range of different sources. The LTLS-FE model then simulates movement and transformation of these nutrients and other parameters throughout the landscape. Within terrestrial systems, we represent processes such as plant uptake and growth, soil erosion, decomposition, and chemical transformations, as well as the generation of surface runoff and drainage. We do this both for semi-natural and agricultural land use. And for agricultural land use, this also includes things like livestock and crop-dependent fertilizer and pesticide application. In urban areas, we have a new sewage model. So our emissions are routed through either septic tanks or wastewater treatment plants before being discharged to rivers or groundwater systems. And our sewage model uses varying treatment efficiencies for different pollutants, as well as sewage capacity, and therefore we can simulate things like combined sewage overflow events. Now, within groundwater, the model represents processes including additional weathering, also decomposition and absorption mechanics, and lateral routing and return flow to rivers. And then finally, within the freshwater system, so the rivers and the lakes, we simulate river routing, algal growth, composition and transformation processes of the different chemicals, partitioning between dissolved and particulate phase, and then settling of particulates to sediment, as well as sediment resuspension under the right conditions. The model operates at a 2-hourly time step and produces national scale water quality estimates across Great Britain, including soil nutrients in runoff and drainage, erosion yields, water quality, most importantly in lakes and rivers, but also things like chlorophyll, total outputs to the coast, river flow temperatures, and other parameters. Next slide, please. Our key challenge in model validation is comparing our outputs at a 5 kilometer resolution against sometimes sparse observation. So here's a list of the observation data sets that we are currently using. Before this data can be incorporated into our validation, we need to do extensive quality control, because sometimes we see things like wrong dates, wrong locations, wrong units. All of this needs to be assessed. It also needs to be assessed whether the observations are representative of the average conditions across a 5 kilometer grid cell. For example, some measurements may have been collected from very small tributaries that are not really representative of the wider catchment average. We also need to consider things like the limit of detection. The limit of detection varies spatially and throughout the years. You can see here on the bottom right, some cadmium concentrations I'm showing median cadmium concentrations across the HMS sites. And you can see that where you see these plateaus, these are level of detections where people fail to detect cadmium, and it looks like you have this sort of downward trend, but really what you're mostly seeing is technology improving and us being able to actually detect cadmium better and better. Next slide, please. I'll now briefly show some examples of some model performance using nitrate as an example. So for each determinant, we compare against observations, and we look at things like spatial percentage bias, which is the model bias versus observation map that we see on the right. This assesses how well the model reproduces spatial patterns, and then also the coefficients of determination, or R squared, calculated from seasonal average load estimates in rivers. And then obviously we look at some major rivers and look at the time series to assess how well we are able to reproduce the observation data. Next slide, please. So here are some additional time series for some suspended solids, some flow, some DOC, and some nitrate. We do this for quite a lot of different pollutants, but here are just some examples to show where the model is doing particularly well. This is showing those values at Thames at Teddington. So this is for a single site. Next slide, please. So as I said before, we do this for more than 40 different determinants, and many of our variables show pretty good performance on average, although it obviously varies between the determinants, and sometimes the lack of data or the lack of data above the limit of detection limits how well we can reproduce the observed data. So I'm showing here a Pearson R coefficient on the top right as a bar chart, where the top, so a value of 1 is very good, and you can see for things like macronutrients and metals, we are doing quite well. For the micropollutants, we are still trying to improve things. But again, this is sometimes down to sparser data. Then on the bottom, I'm showing the ratio of model values over observed values at a given location at a given time. And you can see here the aim of this plot is that we have a ratio of 1. So this is the red line, and this indicates a good match. Ideally, we want the box plot, especially the 25th to 75th percentile of the box plots, to be overlapping with that line. So while some pollutants are not quite agreeing with how we model them, we are doing quite well. And then I'll pass over to Vicky.
Thanks, Nathan. So I'm going to be presenting on the future scenarios that we've been able to make using this model. So just a big cautionary tale. We're still checking and rerunning the future scenarios. So please consider these a work in progress. We're pleased to be able to show you these, but they may change a little bit over the next month or two. So quick reminder of what we're modelling: water quantity and quality, macronutrients, micropollutants, including pharmaceuticals, pesticides, and 7 different metals. But what I want to focus on here are the future scenarios. Here we've got, we're linking both climate scenarios and socioeconomic scenarios. So we've got 4 climate scenarios. They're based on UK CP18. We're using the downscaled version from Chestscape, and we've got 4 representative concentration pathways, ranging from low emissions, 2.6, to high emissions, 8.5. We're also using UK socioeconomic scenarios. These were published a few years ago, and they're adapted and downscaled from global SSPs using stakeholder co-production. As you might be able to imagine, only certain combinations of socioeconomic and representative concentration pathways are even possible. So, for example, if you look on the little table on the bottom right, you see SSP1, which is the green blob, is in fact only really able to be linked with low emission scenario RCP 2.6, and that makes some sense. Similarly, the high emission scenario 8.5 can be linked with 2, but it's always linked, or often linked with SSP 5. So I'll say a little bit more about it shortly. So what are these SSPs? Well, these are all written up in a number of papers, but I'm showing a few summaries here just to sort of give you an overview. The little summaries here are heavily cut down of what's been produced in Brown et al. 2022, but it might give you a flavour of what the scenarios are. So, for example, SSP1 is effectively the green scenario, where you have a fully functional circular economy, a nice, stable and cooperative society. SSP2, that's more of the middle of the road, moderate economic growth, kind of similar to our own present trajectory. SSP3, that's an unusual one. This is dystopian scenario. It shows how increasing social and economic barriers might lead to a fragmented society. SSP4 is a very unequal society, and SSP5 is what's called the fossil fuels scenario, heavy economic growth, very dependent on fossil fuels. Now, these are very well explained in all the papers, but for me, they don't tell me anything about the rivers. So I asked ChatGPT what it thought these rivers would look like. So I fed this these summaries in here, and it produced me these plots. For SSP1, the green scenario, a very nice river with people rowing on the river and walking along. SSP2, the one most like our own, possibly like ours, only we have fewer banners on the river. That must be a ChatGPT thing. SSP3, the dystopian, that's really quite an unpleasant river, I think. The unequal society, you might be able to see that on one side of the river it's quite well-to-do and built up, and on the other side it's quite poor. And SSP5 is a fossil fuel scenario with lots of urban development. So these are great, but also they don't tell you much about water quality, and for that we need a model. So these SSPs come with big descriptions of lots of things, including population, water use, wastewater efficiency, and I've picked out here some of the factors most likely to impact on water quality. So down the side, we have our 5 scenarios. Along the top, we have some of the various factors we need to consider. And as you can see, they go up and down. Each scenario is very different, and even the change in time. So there's a wide range of factors. How do we turn these into emissions of pollutants for our modelling? Well, let me show you. So I'll give you an example. Fipronil, which is the flea treatment. The way we turn these scenarios into numbers is as follows. So we take our set of SSPs, our 5 UK SSPs. We look at our and they scientists work with stakeholders to identify semi-quantitative trends based on for things like population or dosage or pet ownership. And there they try and they estimate for each scenario whether the change in perhaps cats or dogs would be increased or decreased on different scenarios. We link these with spatial trends from things like population density or land cover, spatial information, where we can turn these sort of graphs into spatial graphs. And then we associate the high, medium and low changes with a percentage change, and we press the button and out comes future projections of fipronil loads in wastewater. The graph here is showing time from 1980 to 2080 along the bottom, and up the side we have future projections of fipronil load in wastewater. All of this is assumed to pass through wastewater treatment. For fipronil, we assume there is no wastewater treatment. It's all pretty much unchanged. But in our freshwater model, we assume we estimate fipronil transport, partitioning and photodegradation. So let me show you some of our first scenarios. First up, river flow scenarios. So I'm going to show you a few of these. This is river flow. The dominant drivers of future change in river flow, well, we'd expect it to be rainfall, air temperature, would impact on potential evaporation. And we've also included abstractions and discharges here. As you can see on the left-hand side, I've shown some graphs. These will all be the same. They'll all be from effectively 1980 to 1981 to 2080. And up the side, we have the variable of interest. As you can see, over the 100-year period, air temperatures are projected to go upwards. Abstractions over the historic period assumed constant, then they go down and then up again. Rainfall projections seem pretty constant, but we haven't analysed the trends yet. The end result on annual mean flow projections look like this, which have in many ways a downward trend in most of the scenarios. by the way, what I've done here, I've tried to keep the colours the same throughout all the projections. And for people who don't see colours well, I've labelled them in the order in which they're shown on the graph. So, for example, the top one is the green scenario, SSP1. So most scenarios indicate future decrease in mean annual flow, except for SSP1 on this occasion, but we haven't analysed the trend. Nitrogen is my next example. Dissolved freshwater nitrogen are what we'd expect our dominant drivers of future change will be, well, of course, nitrogen emissions to river, and they will have to include factors such as atmospheric deposition, population and wastewater treatment, agricultural runoff, and runoff from semi-natural land. But of course, we have to take account of changes in river flow because that will affect the concentration. So if you look on the left-hand side, we have a graph of nitrogen emissions to rivers for all the different scenarios. And again, I've labelled them in the order of the colour they are in 2080. SSP5 is the highest at the end, but SSP3 is creeping up in the middle. I wanted to show you, you'll notice the sharp jump in SSP3 around about 2040. We initially thought that was wrong, and then we looked into it and realised that was reflecting the fact that in scenario SSP3, wastewater treatment is assumed to collapse around about 2040, and thus there'd be greater proportion of nitrogen not being removed in wastewater treatment. The end result gives us, these are UK-wide mean river dissolved nitrogen projections, milligrams per litre from 1980 to 2080. As you can see, they look very similar to the emissions graphs. You sort of expect that, and that leap in about 2040 is still there. 3 of the scenarios indicate a future increase in freshwater N by 2050, and a few show a decrease, which is quite nice. So we can also look at these results spatially. This shows a projected change in UK freshwater nitrogen concentration between 2030s, that's a 30-year period, and the 2000s, that's a historic period. Maps that are red show a concentration decrease, and maps that are blue show a concentration increase. So for some scenarios, for example, SSP4 and SSP3, you can see there's blue and red areas showing some areas have a decrease and an increase in nitrogen. So another example, copper. Copper's really interesting. We're showing this because we have to include mine emissions. They're not a huge factor, but they will be for downstream rivers of some areas. Copper emissions to rivers, we can see that for different scenarios. Atmospheric deposition of copper, I'm showing various scenarios here, all for the same period. Total copper emissions to rivers, based on all the estimates, look like this. You can see the biggest emission is for SSP5. Put all this information together and run the model, and this shows our river copper concentration scenarios. Of course, look very similar to the emissions to river, but with some changes. We're also able to plot quality standards for various of our variables. This one's for copper. We've estimated the EQS at 10 micrograms per litre, estimated from WFD value. Next example, bosclid. This is a broad spectrum fungicide. This is an interesting one. For this, you have to take account of changes in arable land cover, how much it will increase in different scenarios. You can see bosclid goes, this is the total bosclid, this is how much goes to the river. That increases from the present day up to about 250 kilograms per year, and then drops back down again for SSP3. The impact on bosclid in rivers looks very similar to the total bosclid emissions. And again, I've labelled them according to the colours they appear at the end. The currently available EQS for bosclid is about 7.2 micrograms per litre, which you'll say, oh, well, that's fine. We're well below that. We are for the UK wide as a whole, but possibly not for particular rivers, particularly not downstream of agricultural areas. Finally, fipronil, our favourite pet flea treatment. Oh, I've gone too far. The dominant drivers of this will be human population, dog and cat ownership, fipronil dose, river flows, and in red I'm showing total fipronil emissions to rivers. Within the rivers themselves, our model projections show that fipronil scenarios will show a modest increase by about 2050, and some with a higher increase. The currently available quality standard for fipronil is 17 milligrams per litre. which again is off the top of this graph, but it won't be for everywhere. And selected rivers and scenarios will probably exceed this limit. What I can show you here we have all our 6 scenarios, and the red areas show where the EQS is by 2050 should be exceeded. It's hard to spot the difference, but you might notice that RCP 8.5, the higher areas are red in the Thames and Midlands. And that's all from me. Over to Steve.
Okay, next slide, please. Okay, so I'm just going to talk a little bit initially about how we're now going on to doing the biodiversity modelling. So one thing that we've done is to develop a chemical mixtures tool. The approach of this is to take the fairly large number of chemical concentrations that we will predict for every time and space point in the model to a smaller number of effect metrics. And to do this, we're using 2 separate models. First of all, we're using a mixture model for metals and acidities, WAM-FTOX, to quantify the overall mixture impact of metals. And secondly, we're using a different type of model, which produces similar outputs, though, an MS-PAF model for the organic chemicals, which links the overall toxic pressure with the relative species richness that we predict. That's all. They're all based on laboratory data, so we're not saying what this produces is going to be absolutely on the button. It is essentially a means to reduce a large set of chemical concentrations down to a smaller number of overall effect metrics that can then feed into the biodiversity modelling. Next slide, please. So now to come on to the ecological modelling. This is all being done by our colleagues at Cardiff University. And the ecological data that they have put together is now being put together with the historical model outputs to understand how different stressors impact freshwater species. And the example plot you can see down here at the bottom left is the model changes in juvenile salmon abundance in time and space using the model simulated climate and water quality outputs from the 1980s up to 2020. And over there here on the right hand side, we can see a couple of charts which map out the percentage change in juvenile salmon abundance at various points for 2 different time periods. So 1984 to 1995, we've been seeing a decline in salmon, and then 1995 to 2005, we're seeing, we'd hope, something of a recovery, or at least not such a bad situation. And this is being done not just for salmon, but also for other fish species, for invertebrates. And our colleagues are also working on producing metrics like trophic diatom index as outputs of the ecological modelling. So next slide, please. So now we'll talk a little bit about our outputs and our proposed stakeholder engagement. So first of all, in terms of disseminating the model, we are going to, at the end of the project, publish the model code on GitHub and make it available so that anybody can pick it up and use it, develop it further, etc. Our new data sets, we are already working on getting our emissions data sets published, and we are planning to publish as many new data sets as we possibly can at the end of the project. We have been producing a bespoke web dissemination tool, which is for users to explore the different different outcomes of the model under the different scenarios, and to download underlying data. And I think in a slide or two we will actually have a hot off the press screenshot of that. And we're also planning later this year, we're planning a couple of stakeholder events to present the outcomes of a subset of the model scenario projections, and to explore how stakeholders, especially people who are interested in developing policy, might interpret and respond to the outcomes of the different scenarios, to discuss potential plans for adaptation, mitigation and detection of risks to the freshwater environment. And there is also potential for a policy brief or a post note, which we could develop at project level or jointly with the Freshwater Quality Programme overall. That is still under discussion. Next slide, please. Okay, so this is a summary of our plans for our stakeholder workshops in the late autumn of 2026. We're proposing to have 2 online sessions. The first session is to explore scenario outputs, is for those people who attend, is to really immerse them in the implications of what might happen under different socioeconomic and climate projections for chemical emissions and water quality, and critically, what are the drivers of those? Are they, for example, drivers that we may be able to manage, or are they drivers that we either need to mitigate or adapt to in some way? And we're going to use a subset of our scenarios. We're going to take SSP2, which is our middle of the road minimal change scenario, coupled with both a moderate and high climate change scenario. And also SSP5, which is our fossil fueled high economic growth scenario, coupled again with a high climate change scenario. So we're going to give stakeholders an opportunity to really get into what's happening to our freshwater quality projections under those scenarios. And then in the second session, we're going to give our stakeholders an opportunity to brainstorm policy options that they might consider to respond to what is happening to water quality under those scenarios. So mitigation, management and adaptation strategies that they might want to think about implementing in response to those changes in freshwater quality, and therefore to take those and to assess their feasibility and their effectiveness under those different SSP and climate scenarios. Next slide, please. Okay, so here is some example output hot off the press of what our prototype web tool is going to look like. So on the left there, we can see spatial maps. This currently only works for the historical model outputs. So we have fluxes and predicted concentrations of copper across parts of southeast England for the year 2020. And then over on the right hand side, and this is actually for a specific 5 kilometers squared, we have copper emission to the river and projected copper concentration in the river. So this is very much a work in progress. As we develop more model outputs, we are going to be adding them into the web tool to allow people to look not just at historical data, as you see here, but also the projections under our future scenarios. So next slide, please. So that concludes our presentation. Thanks to the whole team, not just ourselves at UKCH, but our colleagues at Rothamsted Research, who did the agricultural soil model development and implementation. The guys at Cardiff and Martin at Boburn Consultancy, who are doing the ecological modelling, and our colleagues at BGS, Dan and his friends, who've done groundwater modelling and also helped us out with the emissions of metals from mines. And also thanks to our stakeholder partners, you can see the logos there on the bottom right, who have provided advice and guidance throughout this project and who we hope will be very happy with the outputs. So thank you very much for listening.